Good afternoon, and welcome to the UCSF Academic Senate's 10th Annual Faculty Research Lecture in Translational Science. I'm Margarita Lightfoot, Chair of the Committee on Research and Professor of Medicine, and Chief for the Division of Prevention Science here at UCSF School of Medicine. It's my honor to introduce the Academic Senate Faculty Research Lecture. This award recognizes exceptional career achievement in translational science research. The Senate has two other faculty research lectures, one in basic science and one in clinical and population sciences. Nominations are made by UCSF faculty who consider scientific research contributions of their colleagues and submit nominations for this prestigious award to the Academic Senate Committee on Research. Each year, our committee selects a recipient for this award. I'd like to, first and foremost, to thank that committee for its hard work. Today, we are very proud to honor one of our colleagues, Dr. John Fahy, Professor of Medicine in the Division of Pulmonary and Critical Care Medicine, with the 10th Annual Faculty Research Lecture in Translational Science. Now I am pleased to introduce Dr. Dean Shepard, who will provide a formal introduction of Professor Fahy.
So thanks so much. And it's really my extreme honor and privilege to introduce John Fahy as the 2021 Faculty Research Lecturer in Translational Science. So I've known John for 31 years now. He grew up in Ireland and obtained his medical degree in the equivalent of a medicine internship at University College Dublin, and then the equivalent of a medical residency at Trinity College Dublin and the Federated Dublin Voluntary Hospitals. He worked for a short time as a physician in Dublin, and then came in 1989 to UCSF to do his pulmonary fellowship. He joined Homer Boucher's research lab a year later, and after just 3 years in fellowship, was recruited to the pulmonary faculty at UCSF in 1993. So this is a picture of a younger version of John and a younger version of his research mentor, Homer Boucher, who also turns out to have been my primary research mentor. John came from chilly Dublin, and he was really eager to engage in the sunny California lifestyle, as you can see here by his outfit, and also by one of his first purchases, this used Mustang convertible. And John's posed here at the base of the Golden Gate Bridge on what I assume was one of the few actual sunny days during his first summer in San Francisco. So it's really been a great pleasure to watch the development of John's career. And I think his work has been characterized throughout by always focusing with the laser focus on human disease. So he never engaged in using model organisms or simplified systems. Really, his goal was always to anchor his research in the humans who had the disease he was studying, which has been principally asthma. And although when he started, it was very difficult to obtain samples and to do real research that was informative about basic biologic mechanisms in people, John was not perturbed by that. And throughout his career, he's been way ahead of the curve in developing new tools and taking new approaches to apply each advancement in biology to human airway biology. So I'm just going to highlight a few of the really seminal contributions that John's made, and he's going to talk more about these in detail, I'm sure, in his talk. So one really important finding from John's lab is that his lab was really the first to use genome-wide analysis of gene expression to characterize molecular phenotypes in patients with asthma, and to show that these molecular phenotypes actually had critical effects on responses to treatment. And precision medicine is now almost a cliche, but this was really the first example of true precision medicine applied to lung disease. And I have to say that it changed the way that many of us pulmonary physicians viewed patients with asthma and approached our practice. Because before John's work, most of us presumed that everybody with asthma would respond, for example, to corticosteroids. And we often tended to blame patients if we prescribed these drugs and they didn't get any better. But it turned out, as John and Prescott Woodruff showed, only about half of the patients with asthma actually respond to these medicines at all. And that's really determined by their molecular phenotypes. John also recognized early on that mucus hypersecretion and especially viscous mucus was an important characteristic of asthma and contributed to symptomatic asthma. But at the time, there were really no good tools to study mucus produced by patients. So John developed the methods to characterize the biophysical properties of expectorated mucus, and then was able to use these tools to develop novel and potent modulators of mucus viscosity that he's now been working very hard to move into the clinic. Similarly to this analysis of the mucus itself, John realized that there were no good tools to quantify mucus impaction that was actually occurring in patients. So together with Eleanor Dunnican, they developed precise quantitative methods to use CT scanning to quantify mucus plugging in patients, and has demonstrated the clinical significance of mucus plugging itself. But probably more important than John's contributions to airway science, as critical as they've been, has been his contribution to the next generation of leaders in this field. So at this point, John has really trained many of the next generations of leaders in airway clinical investigation. And I'm sure he'll mention the contributions that many of these have made to the work that he's done. So I'm really very much looking forward to hearing this talk, and I'm sure you are as well. John.
Thanks, Dean, and welcome everyone. I want to thank the Academic Senate and the Committee on Research for awarding me this prestigious lecture, and I very much want to thank Dean for nominating me. I'm truly grateful. UCSF is star-studded, and I'm humbled to be able to receive an award like this when so many of my colleagues are so deserving. I am going to talk today about the opportunity I've had at UCSF to help personalize treatment for asthma. And as Dean alluded to, I came here in 1989, just as the HIV epidemic was really reaching a pinnacle, or it was really reaching its worst. And it's sort of ironic that I'm sitting here today just as the SARS-CoV-2 pandemic has also been a bookend to my career so far. So these two viral scourges have really informed my career here to some extent, and I will be alluding to how both of them have done that during my talk. I'd like to start by presenting three cases very briefly to illustrate the heterogeneity of clinical traits in asthma. That would be typical, as Dean says. I always like to try to stick with the patients in terms of how I organize my research program and even my talks. My lab, though, has developed a focus, and it's been largely in the area of eosinophils, type 2 cytokines, and mucins, and I'll go over that. I know there's a variety of people on the call, including some friends and family, and I will, for that reason, try to make it somewhat understandable. I'll touch on COVID-19 because I think we have to talk about COVID-19 for all sorts of reasons. It's been the big elephant in the room for the last year, and continues to be a source of concern and blight in the world. And then for a career lecture like this, I think I'm obliged to reflect a little bit, and I will do some reflecting at the end. And in order to keep some of you from maybe logging off, I'm going to say that these reflections will revolve around music, butterflies and fish, cooking, and then perseverance and resilience. And if you're wondering what all that's about, it's very short, let me assure you of that. But you're going to have to stick around to hear exactly what I'm going to get up to there. And of course, a career like mine is not possible without many, many people helping me along the way, and I will express my gratitude at the end. The first case I'd like to tell you about is an 18-year-old woman whom I saw a couple of years ago, who had described to me having asthma since childhood, but it had really gotten bad in her teenage years, and she'd been to the emergency department twice and been hospitalized once in the past month. She was super allergic to all sorts of aeroallergens, and her life had been severely impacted by asthma, such that she had quit college after 6 months and was staying home at the time she saw me. She was on a bunch of asthma medications, including a steroid in the form of Symbicort, a bronchodilator, and she was taking prednisone 20 milligrams a day, and she was also on Xolair. Comorbidities included allergic rhinitis and severe urticaria or hives. So this is an example of someone who has very bad asthma in the sense that it's really impacting her life and her quality of life. Her physiology here shows a flow volume curve wherein patients take a deep breath in, going from right to left, and then blast it out hard. And this blue line here shows that she can't hit her peak flow that's designated for her age and gender. And as she comes down the flow volume curve, she's curvilinear. But more important, when she takes a bronchodilator, she has a very large bronchodilator response. So her baseline FEV1 is only 55% of what it should be, and her post-bronchodilator or post-albuterol is 3.16 liters or 85. So when patients have these very large increases in airflow after a bronchodilator, it really indicates very labile airways and is worrisome to me when I see that, especially in the context of a young person who's been hospitalized and in the emergency room, because the asthma is not usually a fatal disease, but it kills about 5,000 people a year in the United States. And there are certain subsets that are at risk, and this kind of patient would get my attention for that. When we look at a simple complete blood count and differential, one of the things that's highlighted in yellow here by the readout is that she has a high eosinophil count of 3,500. And we'll come back and talk about that. So this second case is an older patient, a 66-year-old man who'd had asthma since the age of 40, frequent symptoms, frequent prednisone bursts. He was also on an inhaled steroid, this time in the form of Advair. And his comorbidity was not so much allergic rhinitis, but actual chronic sinusitis with nasal polyps, a rather peculiar problem where patients actually have polypoid growths in their sinonasal cavity and often comes on in adulthood. He too had low FEV1, forced expired volume in 1 second. His airflow was only 50% of what it should be. Had a bronchodilator response that was about 14% here, but he wasn't able to restore his airflow with the bronchodilator. And he too had a high eosinophil count. But the profile of these patients, quite different. The other person, young, having asthma since childhood. This is an older man developing adult onset asthma. And then a third case is this 55-year-old woman who'd had asthma, again in her adult years, taking prednisone a lot, which is problematic for these patients because it gives them so much morbidity. Also having exacerbation proneness in that she's been in the emergency room a lot in recent times and having these problems despite being on a lot of prednisone. Looking at her comorbidities, she has hypertension, taking an antihypertensive, and she has diabetes mellitus. She's also obese, having a BMI over 30 at 34. Again, her lung function being low at 50%. Now, when we look at her complete blood count, her eosinophil count is normal or low, as one might expect it to be on prednisone, just as a point. But what's notable is that she has a high white count and she has systemic neutrophilia. So her profile is not so eosinophilic or allergic. It's more of obesity and metabolic dysfunction, as evidenced by these diseases associated with metabolic dysfunction, which include hypertension, diabetes. Just as an aside, this kind of patient would have been greatly at risk of bad complications of COVID in the last year. So as we ponder this clinical heterogeneity of asthma, the idea is that asthma is not monolithic. It's not the same for everybody. These different colors are meant to indicate that patients have different flavors of the disease. And one gets to thinking, might the heterogeneity of the clinical traits be associated with differences in disease mechanism? And that is what's going to drive personalized treatments. So if all of these patients have a different flavor and they need a different flavor of inhaler, how are we going to know how to sort it out? And one of the problems with traits as a driver of personalized care is that traits are non-specific in their mechanism. Everybody who has airflow obstruction who has asthma doesn't have that airflow obstruction because of the same underlying molecular mechanism. So as I began to develop my career in this area and to pursue what some people like to call translational research, but that's not a term I particularly like. I prefer the term mechanism-oriented clinical research in asthma, because it really makes clear that the patient is at the center of this exercise. And one has to think how to develop and optimize experimental approaches that can be applicable in humans. And I would say I was very much benefited from being in the pulmonary and critical care division at UCSF and in the CVRI, because all of us in that environment were very much directed towards understanding mechanism. And so the question became, if you were interested in mechanism and interested in humans, how do you go about doing mechanistic research in humans? And we can use lung physiology and evoked phenotypes, including aeroallergen challenge and hypertonic saline challenge that I learned how to do in Homer Boucher's lab. We also can apply imaging. Computed tomography lung images are very powerful ways of imaging the lungs. And we can also collect biospecimens from the lungs of patients using induced sputum, which I'll talk more about. And those samples can be analyzed in fairly sophisticated ways, as Dean alluded to, because the last 30 years has been noted for all the technologies that had become available to interrogate tissues. So we can not just use cytology, which was a sort of a standard in pulmonary medicine for decades, but we can do gene expression, we can measure fluid phase measurements, we can do biophysical properties. And in doing bronchoscopy, we can collect endobronchial biopsies, and we can get fancy there too with laser capture, apply stereology for quantification, apply gene expression to brushings, and also lots of things to lavage. And in addition, because it's important to have questions that can be answered specifically, maybe ex vivo, we did in my lab get into culturing airway epithelial cells ex vivo at air-liquid interface as a model of the airway. And so all of this would not have been possible without a tremendous investment in me by leadership at UCSF. And I'm very grateful for the fact that in 2007, Sean Coughlin, Hal Chapman, and Lee Goldman set me up with a very nice research infrastructure, which comprises a clinical research unit on 13 Moffitt, which actually used to be Sam Hallgood's old space, and Sam is now, of course, our chancellor, and the Fahy bench lab in HSE 13. So I have this dual DUAL infrastructure, which happens to be something of a jewel as well. And we have this design for our Airway Clinical Research Center that's quite intentional. And it shows the physician or the healthcare provider interacting with the patient in a partnership to understand mechanisms of disease by incorporating information from the patient in helping us understand what mechanisms we're trying to actually uncover. So back to 1989 for a minute, and the fact that in 1989, pneumocystis pneumonia was a serious problem in San Francisco. And as a pulmonary fellow who arrived from Dublin with my first rotation at San Francisco General Hospital, we were diagnosing multiple cases of pneumocystis pneumonia a day. And this was providing a huge mortality to the HIV positive population in San Francisco at that time, and put a great strain on resources to actually diagnose patients who had pneumocystis pneumonia, because this chest x-ray on the left is non-specific. These patients were immunocompromised, and there were other organisms that could be causing their respiratory failure. But there had been work at San Francisco General just prior to when I arrived, emphasizing that pneumocystis pneumonia can be detected by analysis of sputum. And when I came as a fellow, we would be constantly going up and down to the lab to check to see if the person's sputum was positive, because if the sputum was positive, we had a diagnosis. If it was negative, we had to go on and do bronchoscopy. Patients were left fasting, and we had to deal with that. And I recall talking to Valerie Ng and talking to her about, we would look down the microscope together to see if patients had pneumocystis in their sputum. And what was interesting to me when I was looking at the sputum with the folks at San Francisco General in the microbiology department, they weren't using simple smears, which is what I'd grown up using, where you put sputum on a slide and scrape it and then just stain it. And in that scenario, you can detect gram-positive cocci, as on this left-hand side one here, you can see some cells, but they're not very visible, and you wouldn't trust yourself to tell whether that's a macrophage, an eosinophil, or a neutrophil. But what they were doing at San Francisco General was they were using sputolysin, which is dithiotrietol, and we'll come back to that later on, to homogenize the sputum, and then they were cytocentrifuging it out onto a slide, and you could get pictures like this. Now, this is a slide I made myself with an asthmatic induced sputum. But really, it was night and day. And when I saw this, I said, wow, that looks like BAL. And we could maybe do induced sputum as a non-invasive method to determine whether patients with asthma had certain types of cellular inflammation in their airways. And so we started to use induced sputum to assess airway inflammation in asthma. And we found, looking, that patients had sputum eosinophils. Some had a lot, some had a little, and some had none. And I was quite surprised about the some have none, because at that time, there really was a prevailing wisdom that everybody who was asthmatic had eosinophils, and not just had eosinophils, but had lots of eosinophils. We found lots of people who had no eosinophils. And this was work I did with Homer Boucher as a fellow in his lab, with Jane Liu and Hofer Wang were the technicians that Homer had in his lab at that time. And it was a really exciting time to be working in that lab, and Homer really illustrated for me that you could do mechanism-oriented research, and we had the capacity to do these analyses ourselves, not hand them off to another lab to do the analyses. So I really want to credit Homer for getting me started on this journey. The other thing that we did, apart from just analyzing the sputum for cells, we analyzed it for fluid phase measurements. And eosinophil cationic protein is a soluble marker of eosinophil activation. And so we started to measure ECP using a radioimmunoassay in the sputum supernatant. And I remember doing this with Jane Liu, and I was actually quite impressed that we could do it. And it actually demystified for me for the first time the capacity to do some bench type research in a human sample, because I was essentially a clinician. I had no idea how to do any research. And so getting some confidence that as a physician, you can do science was really important. And we went on to publish a number of papers on sputum, and that process gave me more and more confidence that we could make these measurements. And also more and more awareness that there's a lot of good to be said for keeping custody of your own sample and not necessarily having someone else analyze it on your behalf unless you have to. But another point I wanted to make on this slide is when you look at the eosinophil cationic protein, the average value is clearly higher in asthma than in normal, but there's a good number of people who've got normal values as well as high values. So in all of these analyses, whether it be by the cytological side or the fluid phase side, one can see that there's heterogeneity. I mean, what distinguishes this really high person up here from this really low person down here? And can they all be treated as one in a one-size-fits-all paradigm? Probably not. So my research focus then, as I started to build my own lab, continued to be on eosinophils, type 2 cytokines, which I'll introduce in a minute, and mucins. Dean mentioned mucins, and I've had a long-standing interest in mucins and mucus hypersecretion, going back actually to my training in Dublin. So the research focus involves these type 2 cytokines, which for folks on the phone or on the Zoom who don't know, 3 of them are IL-13, IL-4, and IL-5 are characterized as type 2 cytokines. IL-5 is an important cytokine as a differentiation and survival factor for eosinophils. So anytime you see those eosinophils, like I was showing you, the chances are type 2 inflammation is involved and IL-5 is playing a role. IL-13 is another type 2 cytokine, and one that can activate epithelial cells and change their gene profile and change their phenotype. It can cause these cells to produce more mucins, which are long polymeric molecules, and it can also cause these cells to secrete chemokines like eotaxin 3 or CCL26 that call in eosinophils to the airway. So type 2 cytokines are the effector cytokines of type 2 inflammation, and a prototypic cell in type 2 inflammation is eosinophils and mast cells, which I won't talk about more today, but this sort of little scheme here is what I've spent most of my career working on. And I will say that I've had a lot of support in doing that, and I want to sort of convey some of my gratitude for that as I go along here. I've had tremendous support from UCSF, from the Sandler Basic Asthma Research Center, from the CVRI, which has supported me, and from the endowed chair, the Stuhlbarg chair, that I'm delighted to hold. Michael Stuhlbarg was a dear friend to me for 15 years before he died, younger than he should have. I also want to acknowledge the support that I've gotten from the NIH. I got my first oral one in 1998, and I've had multiple awards from the NIH since then. And that first award, the program officer was Patricia Noel. And to this day, Patricia Noel has been the program officer for all of my grants and has been a tremendous source of support for me, and I think a tremendous visionary and advocate for asthma research at the NIH. So I think the NIH is a national treasure. I think many of us who live here for a long time might forget. I don't forget because I came from Europe and there's no similar structure, and the NIH is just the best. So I don't have time to get into a whole lot of individual mechanistic pathways today, but it's important that I tell you a little bit about interleukin-13, because in work that we did in the lab, and Prescott was very involved in this, IL-13 activates epithelial cells, as I said, and 3 of the genes that are upregulated tremendously by IL-13 are periostin, CLCA1, and SERPINB2. Now, the names of these genes don't really matter too much. What's important is that because IL-13 is present in very low quantities and sort of hits above its weight, you can't measure it easily in biological fluids, but you can measure the signature of its presence. And these 3 genes represent the signature of the presence and activity of IL-13. Now, we did a study back in the early 2000s where we recruited 42 asthmatics and 28 healthy subjects. These were relatively mild asthmatics. They were steroid naive. And we did a study with bronchoscopy where we bronchoscoped them and brushed their airways, the bronchial tubes, to collect these airway epithelial brushings. And most of these epithelial brushings contain these epithelial cells, which actually have a bottle brush look to the top of them there. And when we brush those epithelial cells, we can then subject them to gene expression profiling. At the time, we were using arrays. The Affymetrix U133A was the prototypic gene chip back in 2005, 2006, when we were doing this. And we also took these asthmatics, the 42 asthmatics, and put them onto fluticasone or placebo in a parallel randomized trial. This was a pretty impressive study to pull off as a single center. I give Hofer Wang, who was a member of the PF lab at UCSF for a long time before he joined Homer's lab. And then he worked with me extensively for many years. And Hofer was just an amazing coordinator who single-handedly practically pulled off this study. And it led to really important findings. So when you look at these 3 signature genes of IL-13 activity, because work from the Loxley lab and others had shown that type 2 inflammation and IL-13 was really critical to asthma pathogenesis in asthma models, we were looking at the expression of these 3 genes, expecting perhaps to see that their expression will be upregulated in all of the asthmatics. And this is a heat map shown here, and high expression will be shown in red, low expression is in blue. That's why it's a heat map, I guess. Red will be hot, blue is cold. And not surprisingly, all 3 of these genes, when they came on in the epithelial brushings, came on together. So patients who were upregulated in periostin tended to be upregulated in CLCA1 and SERPINB2. And all of the patients who had that upregulated signature were asthma, as distinguished by these A's here. What was surprising is that there were a good number of A's clustered in among the healthies. So 3 A's here that weren't hot for these genes, and therefore didn't have evidence of IL-13 activity in their airway. So what's going on with them? We really thought that all of these patients, because they had mild asthma and were pretty homogeneous in their clinical phenotype, would be pretty homogeneous in their molecular phenotype, but they weren't. And so 22 of the asthmatics of the 42 had what we call a Th2 high asthma subtype, and 20 had Th2 low. Now, why Th2 high? Well, Th2 is the number 2 subset of CD4 helper T cells. And the helper T cells at the time were considered the main cellular source of IL-13, IL-5. There are innate cells now that are thought to be quite important in this as well. But at the time, the CD4 positive T cell was thought to be the main lymphocyte producing these cytokines. And so Th2 high asthma was the molecular designation given to patients who showed upregulation of these signature panel for IL-13. And of course, not surprisingly, based on what I showed you for IL-5 having a big role in eosinophil biology, IL-13 as well, the patients who were Th2 high had eosinophils in their bronchoalveolar lavage. So in some ways, this wasn't entirely different from what I showed you earlier in terms of the field had sort of known that some asthmatics were eosinophilic and some weren't. But I think the prevailing wisdom might have been, well, if they're not eosinophilic today, they might be tomorrow. And really, they're all eosinophilic. We just have to accept that we can't always find it. There was something about doing it with the gene array and in such an elegant fashion as we did here. And I will give Prescott credit for some of that elegance and also would say that we had very good help from Genentech and Joe Aaron in thinking this through at this time as well. But this was really quite interesting for us to determine. And another point that I would make is that when we then took those patients who were on fluticasone and looked at their response to fluticasone in the context of them being Th2 high and Th2 low, only the patients who were Th2 high had an FEV1 response to the drug. And when the drug was stopped, they came down. The people who were the subtype of asthmatics who were Th2 low did not have any response to the fluticasone, which is a steroid. And Prescott and I were really impressed with this finding, and it really solidified for us that this gene expression profile, which was just taken at one snapshot in time, seemed to be marking a subtype of patient that was responding differently to treatment. And this is really what precision medicine is about. We really should only be giving steroids to these Th2 high people if this FEV1 is, the true marker of disease response. We have to be a little bit careful there because there are other markers like exacerbations and such that we have to sort of curb our enthusiasm a little bit for the clarity that this is telling us. But in large part, this is held up that steroids only really work in about half of asthmatics, and that half is the Th2 high half. So as mentioned, this paper was published in 2009. We had published the PNAS paper that Dean had alluded to a couple of years earlier. We then did another analysis of the work with collaboration from Joe. Prescott and Joe worked closely together. Barmak Modrak was involved in this analysis as well. And basically, we published a paper saying T helper type 2 driven inflammation defines major subphenotypes of asthma. And this, I'll just say it since Dean sort of said it, it was really maybe the launch of precision medicine era for asthma. And it established Th2 high asthma as a molecular phenotype or endotype, and that treatments targeting type 2 cytokines should be selected for type 2 high patients, otherwise they might not work. And I'll show you some example of that. And so we can take these asthmatics here and say, well, there's different colors, but 3 of them sort of sit together and then 3 others don't. And you have this Th2 high, Th2 low. And we went on and working with Rich Loxley and others at UCSF, we applied for a program project grant to explore the biology of type 2 inflammation. And this grant included Prescott, obviously, Erin Gordon, who was in my lab as well and is now running her own lab, Max Seibold, who was training with me here and was also training with Stefan Burchard and is now at National Jewish, Mark Ansel and Rich Loxley are in the ImmunoX program. We decided to sort of try to unpack type 2 high asthma. And in this regard, I would really give my gratitude to Rich Loxley, who heads up the Sandler asthma program at UCSF. I say that I study type 2 inflammation, but I really don't. Rich studies type 2 inflammation and is the world expert in that area. What I do is try to learn from Rich and apply what I can of my knowledge of type 2 inflammation to asthma. And I would not be the scientist that I am today without the learnings I've made from Rich and from Mark. And I would also point to Max as being just a super important external collaborator of mine. I keep giving credit to Prescott here because this was really a defining paper for Prescott. It was really fun exercise for he and I to work on this together, and it felt very meaningful, and it's been cited nearly 1,500 times now. And for a primary research paper that's not a trial or a review, both of us I know are very proud of this. So back to this first young lady who was worrying me, and she was on a drug called Xolair, which is actually interesting because Homer and myself did some pivotal early work on Xolair in our interactions with Genentech in the 1990s. But Xolair clearly wasn't working for her. I mean, she's staying home from college, she's hospitalized, I'm worried about her. She has this really high eosinophil count, 3,500. Xolair inhibits IgE, but isn't working. So might inhibition of eosinophils by blocking IL-5 work? I mentioned that IL-5 is one of the type 2 cytokines that sustains eosinophils. If you knock out IL-5 or inhibit it, you will basically get rid of eosinophils. Now, there are several therapeutic proteins that target IL-5. This is the IL-5 ligand here, and then the receptor. There are drugs that target the receptor like benralizumab, and then there are two drugs that target the ligand, mepolizumab and reslizumab. So might mepolizumab work as a treatment for asthma? If the eosinophil is important, surely it will make people better. So the first study that was done, and this might be described as a pre-precision medicine approach, was does IL-5 inhibition improve asthma controls in patients with persistent symptoms despite use of inhaled steroids? So just getting you ready here, I mentioned earlier that traits are an unreliable predictor of drug response because the traits are non-specific. You and I can both be symptomatic from asthma. It doesn't mean we both have the same type of asthma. Nevertheless, the way all these drugs were being tested up through the first decade of the 2000s were to take patients who have an unmet need, who have persistent symptoms despite use of inhaled steroids. So the treatment, this 2007 trial of mepolizumab. They tried 2 different doses against placebo, picked patients who were symptomatic, put them on the drug versus placebo to see if they would get better. Well, they didn't get better. Even though their eosinophils went way down in their blood, their FEV1 didn't change, their asthma symptoms didn't change. And this was published in 2007, had a very chilling effect on the field because there was a lot of interest in using therapeutic proteins to treat asthma. So the eosinophil group in our community of asthma researchers didn't give up on mepolizumab or any of the other therapeutic proteins, even though it was a little bit of a wobbly period. And people continue to revisit mepolizumab and try to ask the right question. So rather than ask, does IL-5 inhibition improve asthma control in patients with persistent symptoms, the question really should be, does it improve asthma control in patients with persistent eosinophilia, despite use of inhaled steroids? Now, this subgroup could be quite small because they're on steroids, and for a lot of patients with mild eosinophilia, the steroids will inhibit it. So as negative as the previous trial was, this persistent eosinophil group must be quite, just a subset. If it was half the patients, they probably would have seen a positive result in the earlier trial. And Param Nair and Paul O'Byrne is a good friend of the UCSF program. He trained here as a fellow. He's also a fellow Irishman. And Param did a great study to see if mepolizumab might work in prednisone-dependent asthma with sputum eosinophilia. It was a relatively small study. There were only about 20 patients in the study, but the patients who had persistent eosinophilia in their sputum, despite being on prednisone, benefited from mepolizumab, whereas those on placebo did not in terms of exacerbations. So this was the beginnings of the idea that mepolizumab might only work in a subset of patients. And this went on then, there was a much larger study by the Europeans looking at mepolizumab, and this is the Glaxo drug, mepolizumab. And so a lot of these are Glaxo investigators looking at a much larger number. This is 300 patients now, randomizing patients who had to have blood eosinophils before they could qualify for mepolizumab treatment. And in that case, compared to placebo, looking at this survival analysis here, the patients who were on the mepolizumab had much fewer exacerbations cumulatively than the patients on placebo. So this really cemented the idea that if you've got a protein therapeutic targeting a type 2 cytokine, you need to select the population that you're going to try it in. So that first patient didn't get mepolizumab, she got its first cousin, reslizumab. It greatly improved her quality of life. She's back in college. It had a remarkable effect on her asthma control. It restored her quality of life. It got rid of this twitteriness and twitchiness in her airways. This is just from a few months ago, showing that her pre-bronchodilator flow volume curve is restored. Now, we don't always get such a dramatic response as this. But in this case, and in several other cases with other therapeutic proteins, we have been able to offer patients treatments that have really been transformative for them. And these drugs now are used with auto-injectors, patients getting them at home, and really it's been transformative. So what about our second patient, this man who had these unusual sinusitis with polyps? He also had low lung function. He had eosinophilia as well. His problem was that he had some persistent bronchial constriction and airflow obstruction despite bronchodilators. And one of the things we noticed when we looked at his CT scan is that he had mucus plugs in his CT. And when I say we noticed, I mean Eleanor Dunnigan noticed. Eleanor came to my lab from Dublin to work in this area. And she and I started looking at these CT scans. And it was timely because I just joined the Severe Asthma Research Program, another NHLBI program. SARP was doing imaging. I had not really incorporated imaging into my system here at UCSF, but we were interested in mucus. And one of the first questions we asked was, well, can we see mucus plugs on CT scans? And the answer was yes. And then what Eleanor did was she devised this scoring system whereby she took note of the fact that the lungs are really divided up into 20 mini lungs or bronchopulmonary segments. And if you have a plug in one of those segments, you get a score of 1. If you have a plug in 2 of them, 2, etc. So Eleanor devised this scoring system, and we applied it in the Severe Asthma Research Program. Now, the Severe Asthma Research Program involves multiple centers from around the United States, and it involves a longitudinal cohort design. So CT scans are done at baseline and at 3 years. And we had over 150 scans available at each of these times. We did the mucus scoring with the help of Brett Elliker and Travis Henry and Mark Schiebler and David Garada, academic radiologists, Brett and Travis being from UCSF, David and Mark being from other centers in SARP. And if you look at the mucus score here going from 0 to 20, a good number of people had no mucus. And then there were some that had a lot of mucus plugging in their airways. And what was remarkable is that when we looked at them at the baseline and then 3 years later, it was as if we made the same measurement twice. And this is an analysis that Monica Tang did just recently, where we just looked at whether people had no plug at baseline and then no plug at year 3, or were plugged at year 0 and then continued to be plugged at year 3. So patients who had plugging in the first scan almost universally had plugging on year 3. This is a Sankey plot. And if you weren't plugged, you tended not to plug. There was some crossover. And this is brought out here in this Markov model where if you had a mucus plug at year 0, there was an 80% chance that you stayed plugged at year 3. If you had no plug, there was a 75% chance that you had no plug. So it seems like that these patients are the same ones going across here, and there's a susceptibility to plugging and a protection against plugging for whatever reason. And that's very interesting to us and is actually a feature of our program project grant that we're trying to unpack. Of course, the patients who had plugging tend to have much lower FEV1. In orange here, these are the patients who have plugs, and green, the ones that don't. And you can see the FEV1 is 50% in the plugged patients and close to normal in the unplugged. It really makes you wonder if you could make these orange patients green by getting rid of the mucus. That would require a mucolytic therapy. When we look at change in mucus score from year 0 to year 3, interestingly, the patients whose mucus score goes up tends to have a decrease in their FEV1. And if their mucus score goes down, their FEV1 tends to go up. So even though this cross-sectional relationship here can't prove causality, this longitudinal relationship between score and change in FEV1 adds evidence that this could well be a causal relationship, that the plugs are causing the low FEV1. And that has some face validity even without this longitudinal data. But of course, the way to test this is to get rid of the mucus plugs and see if we can restore lung function. Now, what Brendan Huang and Franklin and others in the group have been doing more recently is characterizing the shape features of these mucus plugs in asthma using renderings that Brett and Travis and Kimberly do by putting circles on all the axial slices to allow us to reconstruct the plugs. And when we do that, we being Brendan and Franklin, what we find is these plugs have different shape features. Some of them are stubby, some of them are stringy and long, and some of them are branched. And when you look at the rendering of the whole lung, some of these shape features seem to be the same within a subject. So we get this stubby phenotype and this stringy phenotype. The reason this is important is that one can anticipate that if one was to give a treatment for mucus, say, for example, a mucolytic to the patient on the left here, they might have a faster response to the mucolytic than the patient on the right. And so this ability to phenotype the mucus plugs carefully will be very helpful as we go to deploy mucolytic therapies or other mucoactive therapies in airway disease, because we don't have a great roadmap for how to do that. And anytime we don't have a great roadmap for how to do something in clinical science, it's always a bit nerve-wracking because you run the risk of leaving a good drug behind because you didn't give it the right way. So these plugs might be lysed within a few weeks, but these might take longer. And you need to be incorporating that into your thinking as you design clinical trials. So airway mucus plugs and mucus plug high asthma has begun to really be a major concentration of the lab. And it's really because they're clinically silent. And what I haven't really mentioned up to now is that those patients who have those mucus plugs don't complain excessively of cough and sputum production. They just don't present themselves as having sputum. And so you can't rely on that trait of symptoms to identify the patient. And so for that reason, it's been underappreciated as a pathology and as one that I would describe as a treatable pathology. The other important thing to think about is that a normal healthy lung does not have any mucus in the airway wall, in the airway. We just don't walk around with mucus in our airways. Our airways are kept clean by cough and mucociliary clearance. So this mucus plugged airways is really a peculiar form of respiratory failure. And it can cause airflow obstruction, it can cause symptoms. And in work we've done in COPD that I don't have time to show, Eleanor and I have shown that it can cause hypoxemia, and it's also related to infections and exacerbations. So this is, as I said, a peculiar form of respiratory failure where patients have plugging of their airways with mucus plugs. And given that these airways are necessary to transport air down to the gas exchanging region, this is a sort of an out of bounds phenotype to be having. And the idea would be that if you could get rid of the mucus, you could restore lung health. So that getting rid of mucus is not just for the sake of allowing people to hawk up sputum a little easier. Conceptually, the idea is that they're unhealthy and you're trying to make them healthy. So if we were to treat mucus plugs, we need to know the mechanism of formation of these plugs to have a rational drug development program. And here is when we got into looking at the biophysical features using a rheometer that can measure the elastic modulus and the viscous modulus. Ann Innes was in the lab when I started doing this, and she worked closely with the Muller lab over at UC Berkeley. Susan is a chemical engineer and a president of the American Rheology Society at the time we knew her, so she was card carrying for sure. And I'm not going to go into the details of this today, but it just provides a nice way to quantitatively assess how thick a mucus sample is. And when Leo came into the lab as a technician right out of Berkeley, just came into the lab to work as a research assistant. Leo is a supremely curious young man, and got to know Leo very well and have a lot of respect for him. And he's now doing his graduate studies at Rockefeller. But we did a study initially, fresh from having the knowledge of how to do rheology. We took CF sputum because it's easy to collect, thanks to Mary Ellen and Michelle. And we exposed it to DNase or N-acetylcysteine, expecting that DNase would have a greater effect on the elastic properties, that the reduction with the DNase would be greater than the reduction with NAC. And what we found is that samples of sputum treated with DNase did not decrease their elasticity as much as when they were treated with NAC, N-acetylcysteine. Now, what's NAC? NAC has this thiol or SH group on it. And what it does is it can cleave these disulfide bridges by virtue of thiol exchange. So mucins are polymers that have cysteines in them as an amino acid. And if you oxidize those, you can create these disulfide bridges, and breaking those bridges with N-acetylcysteine serve to liquefy the mucus. We kind of knew this because going back to the sputolysin story from San Francisco General, DTT has these thiol groups. But, so we kind of knew that this was going on, that you could liquefy mucus. But we had, I suppose, thought that there must be something else going on as well. And indeed, we wanted to prove that this was the case. And Leo did some very elegant experiments that I can't do justice to in this talk here. But basically what we learned about pathologic airway mucus gels is that healthy mucus is characterized by polymers that are not cross-linked. They don't cross-link like they do here. And it's the analogy I like to give is that if you have redwood trees and you cut them down and put them into the river, they will flow down the river fine, even if there's a lot of them. But if you cross-link them together, they will clog up the river. So this is known from latex as well. Latex flows out of the tree unless you vulcanize it, which is a chemical cross-linking. So the natural scientists knew that polymers like to move independently. If you stop them moving independently by creating these disulfide bridges, you will create stiff mucus. And we did an elegant experiment, or Leo did, with pig gastric mucin, where we exposed the mucin to DMSO, which is a mild oxidizing agent. And what we can see is that the elastic property goes up without any change in the concentration. And it goes up significantly when you oxidize it. And the amount that it goes up is equivalent to what you get by like a 6-fold increase in the concentration of mucin. And the reason that was important is that the field had really been working on the idea that pathologic airway mucus gels were a function of mucin concentration. And that's still partly true, but a large part of the pathology comes from the cross-linked behavior. And so we wanted to develop a better drug than N-acetylcysteine because we got it to work here, but we had to give it in super high concentrations. And when you use NAC in an asthmatic, it causes bronchoconstriction because it is formulated in a very hypertonic solution. And asthmatics don't like non-isotonic aerosols. So at the time, I'd gotten a grant from the NIH as part of what was called the CADET program, or Centers for Advanced Diagnostics, Experimental Therapeutics, another program that Patricia had championed to try to get novel treatments into the patients. And we had been working based on work I'd done in Dublin on sabbatical with Steve Carrington on looking at glycans that decorate the outside of mucin. So these polymers that I was showing you earlier have a coating of sugar, lots of names for sugar, carbohydrate, saccharide. And when you've got a bunch of individual sugars, they form a glycan. And so mucins are characterized by having a very sugary coat. And Sheena and I in the lab were working on looking at how bacteria like Pseudomonas might bind to these sugars using proteins called lectins. And we thought that multimeric lectins might provide a way in which mucins could be cross-linked by reaching across these mucin polymers and binding to glycans in a multimeric way. And we had thought that, and this was work that we conceived with Steve Carrington and then Stefan Oscarson as a chemist who was involved in carbohydrate chemistry, we thought we could develop fucose and galactose decoys that would interrupt this lectin cross-linking. And we were working on that for some years, and it was actually slow sledding to get the concepts to play out. And then Leo began to present his data around the disulfide bridges. And what we found is that it was much more likely that this cross-linking was a cross-linking related to these disulfide bridges. And because we had carbohydrates that we were using as decoys for the lectins, we asked Stefan, is there any way we could put a thiol group onto the carbohydrate and cause it to break these disulfides? And Stefan actually had no problem doing that. And so I'm just going to run a video here what I'm also describing. So this TDG is a thiol modified galactose structure. And this is a galactose, and it has several hydroxyl groups. And Stefan's lab just substituted in a thiol. And we worked on it in the lab and showed that it's much more potent than NAC. And it's also, because of the sugar backbone, it's very suitable for nebulization and dry powder. And so this is a sample of CF sputum. And when we add TDG to the sputum and leave it just for a few minutes, we left it 12 for the video, but it works within seconds. You can see that it just liquefies the mucus easily. So armed with this, actually, we published a paper in Science Translational Medicine. This was a paper that Leo worked hard on. I was delighted for him that he got this publication. Stefan was involved, Stephen Carrington was involved, Sheena was involved. And we also had help from Stan Hazen at the Cleveland Clinic in figuring out some of this. And this was really important because it led to another program project grant, which was to develop TDG as a novel mucolytic. And this was a multidisciplinary academic drug discovery program that we developed with CRO partners. And so we expanded the group at UCSF. Irina Gitlin joined my group. Bill Raymond joined my group. And Ariana Baum and Xavier did the clinical part of this, and Annabelle was involved as well. But we collaborated with Stefan at UCD and Anne-Marie Healy at Trinity College Dublin. This was back to the future for me. And we also had important partners in chemistry and in inhalation toxicology. And thanks largely to just a great team structure here, and I would give Irina a lot of credit for shepherding all of this. We developed a small library of compounds. We ended up going with MUC31, which is not TDG. We did in vivo studies in mice. We did process scale up and manufacturing, and we've just completed GLP inhalation toxicology and are ready to submit an IND and hopefully get this to the clinic in the next 7 or 8 months. This is just an example of what MUC31, which is our compound, is compared to pulmozyme, just looking at those rheology curves over time. This is pulmozyme, which is an approved mucolytic. PBS has an effect on its own. These rheology experiments are not for the faint-hearted. This is all 20 to 30 sputum samples to accommodate heterogeneity. But the MUC31 consistently against pulmozyme and NAC acts faster and has a longer duration of effect. We also worked with Marcus Mall, who has the beta-ENaC mouse going in his lab. This is a model of airway mucus obstruction, overexpression of the beta subunit of the epithelial sodium channel. These mice developed mucus plugs. We treated these mice early in their lives with MUC31 for 2 weeks, and we found that the pathology was improved and that the mucus volume was decreased. And this was just from giving it into the nose of the mouse. We feel we could even do better if we aerosolize it, but that's for another day. Importantly, these mice do die because this is a pretty strong phenotype and they don't like having their mucus plugged airways. And when we give MUC31, we have the mortality in these patients. Give a lot of credit to Annalisa Adante, Marcus's fellow, for working on this for us. And Anne-Marie has continued to work on an aerosol or on a dry powder version of this, because our real dream is to get a mucolytic into a dry powder inhaler, not just to be giving it by nebulizer. Anne-Marie is an international leader in the pharmaceutics of dry powder inhalers, and she's been working in the grant to show that because these are sugars, they're eminently usable as inhaled drugs. Lactose is very commonly used as an excipient, and our drug is not dissimilar from some of these drugs that are used as excipients. It's just that it's weaponized with a thiol group. And so we've been doing work to demonstrate that it's feasible to give these drugs by dry powder inhaler. So our concept is that a drug like MUC31 would be an inhaled mucolytic that could treat mucus high asthma and restore lung health. And we're excited to be working on that. And that's what keeps us coming into the lab every day to try and make this a reality. And we are taking the asset out of the university in the next short period of time, because the grant is coming to the end in the summer or in the next 8 or 9 months. And we're actively seeking to bring this asset into a company we started, and we'll hope to advance the asset into the clinic in that environment, which is exactly the environment that was set up by these TPPG grants to de-risk academic assets and bring them to a point where they can be licensed. And again, I would give Patricia Noel a lot of credit for setting up these programs and being supportive to the investigators who are successfully funded by them. So if we just go to our 3 cases, then this is a lecture in part on personalized care for patients. That first case that I showed you had eosinophilia. Her asthma came on in childhood. She was not obese. She didn't have nasal polyps. She didn't have metabolic dysfunction. Case 2 was different, only in the sense that his asthma came on in adulthood, and he had these nasal polyps, and he also had severe airflow obstruction that was not responsive completely to bronchodilators. And then the third case was a little bit not the same as the others, because there was no eosinophilia. obesity was involved and metabolic dysfunction was involved. So we have this type 2 high, and thanks to work that Michael Peters has done in the lab and investigators in the Severe Asthma Research Program has done, we know that even within type 2 high, there's heterogeneity. So there's this ramp up of the type of the dose, if you will, of type 2-ness. And Michael has a term for ultra high type 2 that he and Max Seibold have developed, working on gene expression to show that some people are really hot for these genes and others not quite so. So what we find, I think, is that patients who are mild, maybe along the lines of what Prescott and I published in our papers in 2007, a lot of those patients have type 2 inflammation and relatively mild disease that can be easily treated with inhaled corticosteroids. But once you start to get up into more severe forms of type 2-ness, you need a therapeutic protein. And it's really great that we have these proteins. They're really preventing us from having to flog patients with steroids. And then when you get to the top end of this, where you see those mucus plugs that I was showing you, and they're very persistent, and interestingly, they don't seem to be completely cleared by treatment with therapeutic proteins. The jury's a little bit out on that, but we certainly see enough scans now in patients who are taking these therapeutic proteins and they still have mucus plugs. So here, I think a mucoactive drug, I don't think MUC31 is the only game in town. Any drug that's targeting mucus could be brought into the scheme here, but I think this is an important part of the scheme. Now, in terms of this other side, the type 2 low, we're at a little bit of a loss in this area. And that third patient that I showed you is challenging, because I think we have to limit or even avoid corticosteroids in some of these patients, because I'm not sure it's helping them. We need to manage the obesity. In some cases, we need to treat infection, because some of them do have airway infections. And a lot of them have systemic inflammation. Michael is working on this, has published on IL-6 inflammation. Importantly, it's systemic. It's not necessarily, this is not necessarily a side of the equation that's driven by lung-generated inflammation. If you look at the patients, they're all obese mostly and have metabolic dysfunction. That's not a lung problem, but their lungs are affected by it. And so I think we will end up ultimately targeting the metabolic dysfunction, treating the insulin resistance and hyperglycemia. It could be that drugs like metformin or other drugs that improve insulin resistance will help these patients. But this is an area of active interest, and a lot of, this could be as much as half of asthma, and we don't really have great treatments. So I want to pivot to COVID-19. And it's a little bit of a sad pivot, because Vincent, my dad, died earlier this year from COVID-19. And this was him visiting San Francisco 2 years ago. And he was a great traveler and great visitor to San Francisco. And he was 92 when he died, but he was 90 here, and he was in good form, and unfortunately succumbed earlier this year in Dublin to COVID-19. And it's just a reminder from my perspective, looking back on the year, we have to remember that the toll this pandemic took on the elderly. So Dad was 92. If you look at the effect of the COVID deaths in the United States in people over the age of 85, even though people over the age of 85 only comprise about 2.5% of the population, they comprise 30% of the deaths. So this was a pandemic that largely killed older people. 75% of the people who died in the United States were over the age of 65. So my family was not the only one to lose a parent to COVID-19. Many people in the United States lost parents prematurely to COVID-19. And I think that's the sadness of it. A lot of dearly beloved older people in our community died from this disease. I will say on a positive, more hopeful note that the vaccines have been such a tremendous success, and Pfizer, AstraZeneca, Moderna, Johnson & Johnson, I just feel amazed that this happened. And back to HIV, I was also, it took longer, but the HIV story is a story for the ages as well. And the fact that our biomedical community could come together and work with pharmaceutical companies to have vaccines in arms. I got my vaccine before 2020 was out. It's unbelievable, and makes me proud to be part of the biomedical community, even though I had nothing to do with this vaccine success. But my lab did try to respond to COVID-19, and everyone did. You know, all guns were trained on it. I'm sorry for seeming to drop into military aphorisms here, but it was the enemy for sure, SARS-CoV-2. And we were having meetings in my lab, as everyone was, when we all were sent home essentially, to see what could we do. And we knew that SARS-CoV-2 had these spike proteins decorating its envelope, and it bound to ACE2. And Bill in the lab and I were chatting. I had mentioned to him that some friends were taking N-acetylcysteine for its antioxidant properties, and we were kind of saying that's kind of silly. We didn't particularly agree with that. But Bill had the idea, well, maybe N-acetylcysteine could work in other ways, because the spike protein has this structure in part because it has a lot of cysteine bridges, a lot of cysteines in its spike. Again, back to those disulfide bridges that I was showing you in the mucins. This is a cysteine map of the spike protein, including the receptor binding domain, and there's 10 disulfide bridged cysteines. And we began to wonder, might these thiol-based drugs, could they cleave the cysteine and interrupt the binding of ACE2 with the receptor? And Critica in the lab started to work on this actively with Bill and others in the lab. We were interested in particular, for reasons I can't go into now, but with cysteamine and MUC31. Cysteamine is another drug that's used. It's not very good as an inhaled drug, but it can be given intravenously and orally. And we were interested in exploring that. The concept being that if the spike needs this disulfide bridged cysteine to interact properly with its native receptor, if you disrupt that, might you disrupt binding? And indeed, Critica and Jing Jin and others in the Vitalant Research Institute, who we collaborated with, in particular Satish and Graham, we did show that MUC31 and cysteamine could inhibit SARS-CoV-2 live virus infection. On the left here, you can see MUC31. This is a cytopathic assay. You can see that if you don't give the drug, all of the cells, 100%, die when you infect them with the virus. But when you go up in the dose of MUC31, you can rescue the cells. Similarly with cysteamine. Now, the IC50 was in the millimolar range, and people were pessimistic that we could achieve those doses. But in fact, you can achieve millimolar concentrations in the lung, especially if you give it by aerosol route. So because we've been working with Lovelace in our drug development program, and Lovelace had the Syrian hamster model of SARS-CoV-2, we did an experiment to compare MUC31 given by aerosol with cysteamine given intraperitoneally in hamsters exposed to the virus. We could only give it by aerosol for 3 days because the animals couldn't tolerate longer. We could give the cysteamine for 5 days. And what we found is that the inhaled drug, MUC31, which was achieving millimolar concentrations in the lung, decreased viral titer, whereas the cysteamine didn't. So then we didn't expect the cysteamine to have any effect on the SARS-CoV-2, the COVID-19, but actually both MUC31 and cysteamine decreased total cell counts in the lavage and decreased neutrophils and macrophages. So even though cysteamine wasn't having antiviral effects, it was having anti-inflammatory effects. And this led us to think that thiol-based drugs have multiple effects, including anti-inflammatory that are independent of an antiviral effect, and got us thinking that these thiol-based drugs are maybe underappreciated for their efficacy and maybe have potential for broader medicinal utility, both as mucolytics, antivirals, antioxidants, and anti-inflammatories. And not all of them are deliverable by aerosol. Not all of them are very potent, but I do think that these drugs could have utility in lung disease, especially since so many lung diseases are associated with oxidative stress, and since oxidative pathways are so involved in a lot of infection responses. So that's the end of the science part, and I do have a few reflections. I promised to decode these 4 different things here. In terms of music, I'll also talk about butterflies and fish, cooking, perseverance, and resilience. I think music for me, it really gets to a comment that Niall Roach, my classmate from Dublin, who's now a rheumatologist in the East Bay here in Ireland. It's really a reflection on the privilege of being a professor of medicine in a place like UCSF. and the gratitude I feel for a career that allows for both creativity and leadership. And it was a comment Niall said to me one day about my work. He said, you seem to be writing the music that other people play. And that sounds a little haughty or maybe arrogant, but you know, on days when you need a little haughtiness or arrogance, this is a thing that can be thought of. And it reflects really on the fact that in the world that we live in science, where the impact of the work that we do daily can radiate out from us and can have impact beyond you know, a single patient. And I think the writing the music that other people play is actually not a bad way to think about that on the days that people might be feeling a little low energy. Butterflies and fish. So again, this is a reflection on the privilege of training young people. And Mara Lakowitz-Scroggins, who worked in the lab for many years and was just a joy to have, and is now a program officer at the NIH, gave me this gift when she left the lab. It's a framed butterfly, an actual butterfly. And she commented in some words she spoke at my house when she gave me this, that you know, she came into the lab as a caterpillar and left as a butterfly. And so that's nice. That was very nice for her to say that. So it's a reflection on privilege, and I have all sorts of privilege as a white male, but honestly, the privilege that I think about a lot is working with young people and helping them to become scientists, and they're helping me to do what I do. The other part of academic life that's really special is the privilege to make friends in all sorts of places. And I have met people all over the world: Japan, Germany, Italy, Europe. Bart Lambrecht is a great friend, and he happened to visit my lab in the summer of 2019. His lab is in Ghent in Belgium. And Bart's a very keen fisherman, and he said, we got to go to Hat Creek. It's one of the 10 great places to fish before you die. I had no idea. I don't really fish, but Bart arranged it. Off we went to Reading, and we had a great day. And I think of all the great days I've had with so many people around the world that are really dear friends. And academics has given that to me, because I'm not restricted geographically in the people that I meet. And it's just a great privilege. The other reflection would be just cooking. I remember watching the cooking channel with the kids, especially in their early teenagers. It was just such a safe thing to do. Nobody seemed to mind switching on to the food channel, watching Chopped or whatever. And what you learn is that great chefs have a point of view that informs their food creations. I mean, you see these chef adjudicators, you know, judging people who cook. They wanting them to have a point of view. And I had thought that, I guess I hadn't thought about it, but I thought food was just a menu. You just follow a recipe. But it made me realize that that's not true, and that great chefs obviously have a point of view, and that informs their creation. And I think that's true of scientists as well. The importance of a point of view. And you have to develop a point of view in order to have a lens through which you look at the problem, and then just a way in which you approach it. And over the years, what I've tried to do is avoid writing reports and write stories that are sort of grounded in that point of view. And the other thing I'd say is it's important to develop some attitude and to enjoy it while you're doing that, you know. And so over the years, I've started writing pieces where I like just sort of leaned into it a bit, you know, had papers that I was, you know, I was trying to convey in the message of the piece in the title. And this one here was actually probably a little bit bad-tempered, a lazy term of uncertain meaning that should be abandoned. But I am particularly allergic to that term, allergic reactive airway disease, which some people who've worked closely with me know. Wrote that with Paul O'Byrne, who was similarly irritated at the time. I would say it's the single biggest reason I get emails out of the blue, because every mother of a child who Googles reactive airway disease gets this and then instantly emails me. So on perseverance and resilience, I think I've tried to learn how to be resilient. I think it's not something that we're born with. And I think it's good to understand how you develop it. And in the science world, it's particularly important because it's a tough, very competitive field. And what I've learned is that I think you need to focus on a problem in order to be expert in that problem. And then you want to be expert so as to be confident. And then you need to be confident so as to be resilient. And Dad sent me this a few years ago. He was in his 88th years. He was getting parsimonious with his birthday cards to John, keep going. I think he kind of knew this, and he knew that it was occasionally good to sort of say that to me. So gratitude, those of us who run large programs and try to do ambitious things, it's impossible to do it by yourself. You need to be surrounded by your institution, support, colleagues, administration, family, friends, and the team. And it's everybody together. And I have, take away any one of these things and I'm not doing what I'm doing. Orla, my wife, is my chief supporter and support, and she has been that for 33 years, and I'm just very lucky. I mentioned my dad, my mom too. I think I get my drive from my mother. She was an amazing person. And my kids, Owen, Emma, and Claire, just a constant source of joy and pride. And we're so lucky. We're so lucky to live in. I got another sunny day here, Dean. So I think we badmouth the weather in San Francisco too much. I've had so many lab members, I can't list them all, but my current lab, I would like to recognize, including Ariana, who's my research manager on the clinical side, Annabelle Charbot, who's the lab manager on the bench side. I think I've tried to mention a lot of these people as I've gone along here. Irina's been key in the program project grant. I'm mentoring Monica Tang, and I continue to help Michael out as a mentor. People I can't do my job without. Gavin Wren is my pre-award person. Lena Yu is post-award. Clarice Estrada I've known Clarice for 25 years. She was the division administrator in pulmonary, then at CVRI. Now she's gone to Dan Lowenstein's office. I'm putting her in parentheses here because I'm not sure what to do without Clarice. So I might occasionally call her up in Dan's office. Key collaborators, in the local environment, Prescott, Aaron, Rich, Mark, and the radiologists that have helped me, and the ENT surgeons that I didn't have enough time to get into the work we're doing together. Max I've mentioned, Stefan I've mentioned. I mentioned Mirish Fitzgerald, who was the professor of medicine at UCD when I was a medical student. I worked for Mirish as an intern and as a medical registrar. You can't be what you can't see. And what I saw in Mirish was you're just a high quality academic scientist and a physician. I'd never seen that before. And that's where my inspiration came from. There at the beginning and since has been Homer. Homer is not just an academic colleague. Homer is a dear friend. I've had multiple dinners in Homer's house, multiple Thanksgivings. He was kind to my parents, my children. Steve Lazarus, just been there since the beginning too. Shoulders to cry on. And staunch supporters, Dean, Bob Wachter, Brian Black, CVRI director, Bob Wachter, Department of Medicine. And I've just been blessed by having strong quality leadership in the institution that I've worked in. And without that, I think I wouldn't have been able to do what I do either. So thank you for your attention. I hope I haven't gone on too long, and I'll be happy to take any questions.
Dr. Fahy, I don't know if you can you see me? We're supposed to be sharing screens. Hi. I wanted to thank you for a really fascinating and really inspirational lecture. And I did want to say, I'm so sorry about your dad. And I'm so sorry for everyone in our audience today who lost people to COVID in the last year. I'm going to pass on any questions the audience has into the Q&A box. I have a question. I was wondering whether the asthma phenotypes you were describing are consistent across different demographic groups and independent of, or if you consider environmental factors in your work?
I would say that we don't consider environmental factors maybe as much as we should. We do definitely collect information on environment, and it wouldn't be a particularly strong suit of the work I do. It's not something I'm expert in using the instruments that collect information about environmental exposures. I do think they're likely important. I find myself, because I run the severe asthma clinic, I do think there's a little bit of a tendency for some patients to overestimate the importance of, like, they think they may have a lot of molds they're exposed to in their houses or something like that. In my experience, unless it's a very extreme mold exposure or something like that, it's much more likely that the problems they're experiencing are not specifically a result of environmental exposures like that. Now, writ large, on a population basis, things like there's good data that people living close to freeways and exposed to diesel exhaust fumes, and there's a big interaction between that and disparities in terms of people's poverty levels. All of that's important. The other environmental exposure that I find myself talking a lot to patients about is cats. Patients love their cats, and cats for some patients are just a source of chronic asthma morbidity. And so I've learned to ask the age of the cat and gently suggest that maybe the cat not be replaced when it passes along. That's the best we can do. But there's a longer answer to your question, Penny, that I think might take too long to answer here in terms of the role of environmental exposures, because for sure the answer is yes, but it's as with a lot of these things, quite complicated.
A question from Param Nair. And the question is, would you please comment on your thoughts of how relevant IL-13 is in relation to the mucus production in asthma, and if this is linked to airway hyperresponsiveness?
Yeah, Param, nice to hear that you're on the webinar. Param is up in McMaster, is a dear friend, long-term colleague. The mechanism of the mucus plugs for sure has to involve IL-13, I think, for all the reasons I was alluding to in my talk. Its effects on epithelial cell function and the role in eosinophil accumulation in the airway. What was the second part of the question, Penny, about whether?
Whether if this is in relation to mucus production in asthma, and if this is linked to airway hyperresponsiveness.
Yeah. Well, there was a nice paper in Nature Communications in January showing that if you get rid of mucus plugs using mucolytics, you improve airway hyperresponsiveness. So I do think that there are mechanisms by which luminal impaction with mucus can affect smooth muscle that I think it'll be interesting to look at that when we do human studies and we find ways to get rid of these mucus plugs and see what gets better. Is it just airflow, or do we improve airway hyperresponsiveness as well? It's conceivable. You wouldn't think of it initially. You would wonder, why would reactivity of smooth muscle be influenced by mucus plugs? But I think the mucus plugs are complicated, long-lived structures. They may be a scaffold for cytokines or growth factors that can have an impact on airway smooth muscle.
Bart Lambrecht asks, do you want to proceed with MUC31 in COPD and CF as well? What would be the ideal patient?
Yeah, thanks again. Bart is the fisherman and obviously a really outstanding immunologist. Any mucus-associated airway disease, Bart, would be amenable. I think we think of this as a final common pathway where oxidative stress from whatever source, an eosinophil, a neutrophil, smoking, what have you, CF, we think the mucolytic should work. And we published a paper just a month ago in COPD showing that there's a subset of the COPD patients who have limited emphysema and a lot of mucus and have airflow obstruction and hypoxemia. And that's a population that I think I would love to treat with a mucolytic to see if we can improve their health, because there's a lot of therapeutic pessimism and nihilism in COPD when you see very sick patients. Can we get them off the oxygen? Can we improve their airflow? And I think there's a subset of those patients that I think could benefit from a mucolytic therapy or a mucoactive therapy. It doesn't have to be MUC31, obviously. I suppose it was obvious from my talk that we have intellectual property and other interests in MUC31, so people should be aware that I have commercial interests in that molecule, as does the university.
Another question is, an anonymous attendee asks, putting together two parts of your talk, it seems that the question as to whether asthmatics, in quotations, are more or less susceptible to severe disease from infections by SARS-CoV-2 cannot be answered without considering the phenotype of asthma. Of the three patients you presented, might the first two be less susceptible, but the third more so?
Yeah, that's very perceptive, and that's true. I think that's absolutely true. There was a lot of concern that asthma would be really a risk for severe outcomes of COVID, and there was multiple papers during the year that sort of said that wasn't looking to be true. But that last person I described who had the hypertension and the BMI issue and the older age, all of those are independent risk factors aside from asthma. And I think when you add the airflow obstruction on top of the hypertension and the diabetes, that would be a bad combination. So I think you're right. The heterogeneity of asthma gives rise to heterogeneity of risk to bad outcomes from COVID.
I should just mention that these people are also telling you how great your lecture was. And Bart says, so proud, I learned you how to catch a trout and identify a dendritic cell, and so grateful of my time in your lab. Anyway, I hope that you get a copy of these questions, because interspersed with the questions is a lot of appreciation of your work and you.
I remember when Bart was caught, I caught no fish in Hat Creek, but Bart caught several fish, as did his son Maxim, and they gave me a fish so that I could have a picture taken. And I've never admitted it, but I would admit it here that the fish people saw that day that I texted around was not my fish, it was Bart's fish.
Confession. Mara Lakowitz-Scrogg is here. She says, congratulations on your lecture award and thank you for that wonderful update and tribute. I think the anti-inflammatory effect of the thiols you presented is compelling from your recent COVID work. Are you pursuing this in the context of chronic infections, i.e., would these drugs potentially have dual benefit in the CF community who suffer both from thick and persistent infections?
Possibly. I mean, I think as a field, the thiol drug medicine field has been sort of upset a little bit by bad versions of those drugs, number one, and some of them are, and not a very organized principle for how they're used. The best example is N-acetylcysteine used for rescuing patients who've had Tylenol toxicity, where everybody agrees it's life-saving treatment. But some of them aren't very potent. They don't have good oral bioavailability. They can't be given as inhaled drugs for a variety of reasons. But there are about 15 of them out there, and there are more coming on stream. And I think that over time, they may come to be viewed as a sort of a steroid-like drug, except they're not steroids. In other words, they have these, I don't know if pleiotropic would be the right word to use in this non-genetic context, but they have broad-based effects as antioxidants, disulfide cleaving drugs. So I think, Mara, the long answer is it'd be interesting to see if some of the COVID work, and we're not the only ones to suggest that some of these thiol drugs might be effective in COVID, might prompt some reconsideration. I mean, these would be some of the hopefully positive things that might come out of COVID, is that they've shaken us all up in a way that we need to think about things a bit differently.
Rick Loxley says, great talk and very inspiring. So another phrase tossed around in the field, where is the capital H hygiene, capital H hypothesis?
Well, that's a question for Rich Loxley. I mean, where the hygiene hypothesis, for people who don't know, is there's been this puzzlement about why sort of maybe the first kind of patient I showed you, the person who develops asthma in childhood and has associated like other diseases, they often start with eczema and then develop rhinitis and asthma, the so-called atopic march. That disease is much more common in Western countries with good hygiene, like patients in sub-Saharan Africa and parts of South America and poorer parts of the world generally don't tend to have those allergies and eczemas and that type of childhood asthma. And there's been a wonder if cleaning up our environment, you know, smaller families, more antibiotics, et cetera, lack of exposure of your sort of nascent immune system, is that the reason why Western children in privileged environments of economics and wealth have a lot of these asthma phenotypes? I think the reason we haven't been able to, there's been some attempt to study that in large scale studies, but because you're talking about young children, it's hard to do an experiment to definitively look at that. In terms of UCSF, some of the people who are taking it on will be Susan Lynch, because some parts of it relate to, you know, gut microbiome and how your gut microbiome informs your innate immune development, or your immune development, innate and adaptive. And so I think where that stands, where it's possible that we will learn that certain kinds of microbiome manipulation in young children could be protective. That would be my prediction for what could happen within a 20-year life period of time. It's not something I'm particularly working on and don't have particular expertise, but that's what I would predict.
Xiaopeng Yuan says, very inspiring talk, John. Missed the lab. Curious about if something like ketogenic diet would help the obesity-related or Th2 low asthma.
Yeah, it'd be nice to hear from Leo as well. All these lab members, thank you so much for tuning in. Honestly, I think one of the things that Michael and I, and this is an area Michael Peters is growing his program on, but I think we've underestimated the effects of obesity on asthma. We had thought of obesity as just having sort of mechanical effects on the airway, but clearly a subset of obese patients have multiple metabolic derangements. And it's known that those derangements are associated with diseases of the heart, of the brain, smooth muscle, you know, skeletal smooth muscle. Well, as a discipline, pulmonologists hadn't really thought of systemic perturbations that are brought on by obesity as having effects on the lung to change the lung's function apart from those mechanical effects. We sort of were rooted in this idea that lung disease comes from the environment and is sort of lung-centric and not outside in. So I think as we begin to develop treatments for various complications of obesity, we should see what effect those treatments have on lung function, whether it's through ketogenic diets, as Leo's suggesting, or what have you. It's hard to know. I have noticed, you know, I run the severe asthma clinic. When patients have bariatric surgery, they get remarkably better from their asthma. I mean, it's just undeniable. And with the obesity epidemic in the United States at the moment, this is what we see a lot of. We see a lot of patients with morbidity from asthma, from obesity, even though we're always thinking about allergies and all the rest of it. But the way the United States is growing literally, and this is what's going to challenge us in the clinic. And I think we're going to have to find a way to manage weight, or else our lung health is going to just get progressively worse. And those patients are supremely susceptible to bad outcomes from infections, not just COVID, but they don't do well when they get pneumonias. They die at a higher rate.
Alex Hansen asks, in the ideal outcome of a dry powder mucolytic inhaler, do you have any sense of the duration of the therapeutic effect? Would this be a multiple times daily treatment, or would the effects be more durable?
It's hard to know. You know, as I mentioned, I think earlier, our only roadmap on this is pulmozyme given to CF patients. And in those scenarios, you know, with CF, it was viewed that you break up the mucus and it comes right back at you. It's a very prominent phenotype when you've got the CFTR mutation. These plugs that we see in these patients, if we lyse them or some other therapy gets rid of them, how quickly do they come back? Such that you have to guard against that by daily treatment? We don't know. It's one of the reasons why I'm pleased to have the imaging as a way to monitor and explore some of these therapeutic efficacies, because I think the field needs to learn. Anybody who says they know whether it should be once a day or twice a day or what have, is only guessing until we get some data. So that's the best I can do with that at the moment.
Anne-Marie Healy does not have a question, but says, thank you, John, for a great lecture and for acknowledging my small input. Wonderful to be collaborating with you. And Steve Carrington says, congratulations on a fantastic talk and very sorry to hear about your father. And he's proud to be mentioned as a contributor, and sent you an email and says he's got to catch up sometime when you're over in Ireland.
Thanks, Steve.
Let's see. Stephen, and more accolades. Stephen Hayes, it's telling you what a phenomenal story you've told today, and congratulations. And says, I've learned much from you over the years, not only in science, but academics and life. Thank you. Ying Wei says, congratulations for the award. I was moved by your reflections. Thank you for a wonderful lecture. Walter Finkbeiner says, dear John, excellent talk. You're writing a great story. Best, Walt. Helena Agarter, hi, John. The mucus casts, in quotations, you are getting from asthmatic patients are beautiful. Do you know what coincides with the different shapes, eosinophilia / duration of asthma, crystals?
Yeah, Helena is a postdoc with Bart and was in the lab for a while. I know she's very interested in crystals, so of course I'm going to say crystals are involved when Helen is asking the question. You know, we don't have an, you know, all we can say, Helena, about these mucus plugs is there's a very strong association with eosinophils. Like I showed you that people who have mucus plugs continue to have them, people who don't have them continue not to have them. The biggest risk for having them is eosinophils. The biggest association for not having them is not being type 2 high, which is sort of surprising. You might have thought that if I told you that the heterogeneity of asthma is associated with mucus in all of the different, like you could get to mucus from a number of different places, but these non-type 2 people seem to be protected from having, they don't, you know, they seem to retain the ability to cough up mucus and not have it plug your airways. As I said, it's a peculiar form of respiratory failure. I use the word peculiar advisedly because it's anti-teleologic. I mean, you're not meant to clog off your airways that are bringing oxygen down, right? So there's a lot of defenses to stop that happening. So it's a kind of a weird thing that it plugs it. And I think the reason we've developed it probably over the years was maybe to sort of trap worms or something, that was the idea behind thickening a mucus cast, was to trap things. That's the best reason I can come up for why type 2 is peculiarly associated with casts and plugs. But you and I need to do some more work on the crystal piece, Helena. Happy to do that. I'd be surprised if they weren't prominent in a lot of these plugs. And it could be that some of the longer ones are more crystal laden than some of the shorter ones. That's possible. We don't have data for that yet.
Neil Roche says, wonderful lecture and great work, John. Well done, Neil. And Michael Peters says, John, you've been the unique researcher who has not only contributed to the science, but has also mentored a generation of scientists who are now global. How does one accomplish this? He's talking to a real softball here.
Yeah. Well, I think part of it is environment. You know, one of the things that I would say about I would give a shout out to the pulmonary and critical care division at UCSF. it's an extraordinary division. Not all divisions are like our division. And we've had a training program that's had fellows since the late 60s, mid 70s. So you come into our program and there are 6 or 8 fellows coming in every year, and we're affiliated with the CVRI as well. And I would honestly give a lot of credit to the CVRI. I liked the setup where the pulmonary division was well rooted in the Department of Medicine, but also had a big association with the Cardiovascular Research Institute. And the Cardiovascular Research Institute had a long history of training fellows from around the world, especially from Europe and Asia. And so there was a lot of people came into the pulmonary division through the CVRI's sort of background. And then in terms of the fellowship program, like people like Michael, who asked the question, came into my lab as a result of being in our clinical fellowship program, which is very academic, very focused on training research fellows. And MDs, I think, like the idea of deploying some of their clinical skills in the research environment. They don't want to start from zero. You know, if you go into the bench lab, you're starting from zero. You're competing with people who just know bench, and so you have to learn all those skills. So I think if you kind of meet yourself and know yourself, one of the things that I think you need to demonstrate is passion. If you have it, there's no point suppressing it. I think it's hard to suppress passion. I think it can be infectious. And so I think part of it is you infect people. And I also think it's important to be very mindful when you're presenting your work, to be very thoughtful about how you do it. I mean, if anyone thinks that this lecture has been good, it has probably taken me about 40 hours to prepare this. Like, this didn't like cobble this together. Why do I bring that up? Well, sometimes you people get inspired by your lectures, and sometimes, you know, you meet them. Like, I remember meeting Bart, I think one time in Madrid or Barcelona, where we were back-to-back speakers, and we weren't that familiar with each other's work. So you just never know who's listening and who's watching and who might want to join your group. And so I think never take anything for granted. I would especially if you're a young person, never take any opportunity to present your work without really preparing hard for that. People form opinions very quickly about you. And so whenever you show your stuff, you know, think hard about the best way to show it.
Mm-hmm. Prescott Woodruff says, wonderful talk, John. Thanks so much for all your support over the years. I'm proud to be part of the UCSF ACRC team and very grateful for your leadership.
Thanks, Prescott.
And Tom Fahy says, John, fantastic talk. As a cousin and friend who trained with you in Dublin in medicine in the 1980s, I'm so proud to hear of your achievements, particularly your mentorship. UCSF has been a wonderful place for you to work as a clinician scientist.
Yeah, Tom is professor of general practice in Dublin, one of the few professors and chairholders in general practice in Dublin, actually. It's interesting, not every medical school has had one, and Tom has himself been a very accomplished academic. He has an E in his name, which always confuses people, but he is my first cousin.
Well, there are no more questions or comments past him, so I think that we've reached the end of the Q&A. I just wanted to congratulate you again and thank you on the behalf of the Academic Senate and the audience today. It was a really fascinating talk, and as everyone's saying, very inspirational, and thank you very much.
Thanks, Penny, and thanks everyone for your time and interest.
Bye.
Bye-bye.